Thank you. Thank you. So, hi. I hope you guys are settling in okay, getting used to 110% humidity around here. So, this is Jeremy. So, Jeremy and I will be covering the topics of genome sequencing, assembly, and annotation. And we're going to do a really high level because we were told that you guys have various backgrounds. Some of you are doing drug modeling and stuff that are not very close to genomes. And so before we get into genome assembly and annotation, I just want to show you how you get acquired sequencing data to do the assembly with. And there are several sequencing technologies that had come out. The first one I'm sure you're all familiar with, the Sanger sequencing from the textbook from your undergrad. We'll review the basic of Sanger sequencing before we move on to the second generation sequencing, which there are a couple of platforms today. The most popular one being Illumina. There's also Ion Torrent and MGI. I will pick Illumina to cover because that's what most people use. And finally, the latest technology, the third generation sequencing, the PacBio and Oxford Nanopore. We'll cover the PacBio since it came out first, and the read accuracy has improved a lot on that platform. And so, all right, let's start with Sanger sequencing. So going back to the undergrads, this is what we've been using the longest of all sequencing technology. It's sort of a gold standard, but now some people have moved the gold standard to Illumina. To do this, we will need a couple of components. So first of all, you definitely need the template that you're going to sequence. You need the primer. This is going to be a chain terminating dideoxynucleotide technology. So you're going to need two types of nucleotides. The normal DNTPs, which is unlabeled, and the special ones that it's fluorescently labeled and also has no hydroxyl group. And so basically whenever the fluorescently unlabeled dideoxynucleotide is incorporated, the chain is automatically terminated. And you mix these as a certain ratios that once in a while, these gets incorporated into the chain and then it got terminated. And so you will have like a very, very short product. And then hopefully at every single base, something get terminated along the way. And once you have these whole product, you take them and you run them on acrylamide gel. And that's like a really, really old way. Most of you are probably not familiar with acrylamide gels. The newer technology turns the acrylamide gel into the tiny capillary, and you can just load the product onto the capillary, which has a camera at the very end. So when it flows, the smallest one will flow first, and then you just read out the colors. So each of the base is associated with different fluorescent dye color. And what you get is a chromatogram that looks like this. And so this is the smallest product, and this is the larger one. So you're probably familiar with this graph. Next, we'll go over the next generation sequencing or the second generation sequencing. So the difference between the Sanger sequencing will give you a read length of the sequence of about 900, maybe 1,000 base pair. You could do, I don't know, 96, 384 wells at a time, but that's sort of like the limit of the number of sequences you could do at once, which is very low compared to these. These you could do millions and millions of sequencing simultaneously, and so they're being done in parallel. There are several platforms, or the three most popular ones being Illumina, Ion Torrent, and the MGI sequencing. Today we'll go over the Illumina sequencing, but if you or your lab happen to use some different platforms, you could look them up. The idea, it's pretty similar. And so the general idea is for these, you have to build what's called a library, which is a collection of small fragments that you're going to sequence. So the first step is to build the library. This is the overview. And then there are some sort of step that will amplify that one piece of DNA into clusters of DNA of the exact same sequence so that you can sequence and capture. And so most of these use the fluorescent dye. The reason that you have to generate cluster of sequence, cluster of templates It's because the camera that is used to detect it is just not sensitive enough to detect fluorescent dye given off from just one molecule being sequenced. So most of these will have some common ground of some sort of amplification. For Illumina, they use what's called a bridge amplification, which I will show you in a little bit. For ion torrent, they use what's called emotion PCR, where you put the template on the bead and then it gets amplified on the one bead. And for MGI, they use a different technology called the DNA nanoball. But the idea is the same, which is to amplify the template. And the third step is the actual sequencing, where you have the fluorescently labeled nucleotide. And this is very similar to the Sanger sequencing. We'll go over this at the end of the Illumina. So just to go into a little bit more detail of how the library is usually made. So you start with your genomic DNA template, and then you break them down into pieces, smaller pieces of the size of the library. For second generation sequencing, usually the size is a couple of hundred base pair. It's very short. So you can break it physically by sonication, or you could do it enzymatically. And after you break the DNA, usually the ends is damaged, and so you have to fix it. You do the repair, and you put on the adapters. And these adapters have priming sequence for the primers that can also contain barcodes or index, which you could use to multiplex samples into one run, because the data output that comes out of these platforms are huge. You could combine several samples into a single run, and then you put a barcode with the unique sequence on each of the samples, so you can track back which sequence came from each sample. After you generated the library with the adapters, you just do a QC check, make sure that the size is correct. If it's too big or too small, the sequencing will not come out good. And then you go through the amplification step, and this is the bridge amplification, which I have tried to explain with the pictures several times, and it failed miserably. So I'm going to show you a video after this of how the bridge amplification works. So we'll skip this. Just take my word for it that after this, you turn one strand of template into clusters of template, and these clusters are going to be sequenced on the flow cell. Finally, we get to the sequencing step, which I'm going to show you in detail on this slide. And so this is the template that you're going to sequence. The blue dots here are the primers, and the nucleotide are being flown in one cycle at a time. Each of the nucleotide has a different color label, just like the Sanger sequencing. And so the special character is that it's got like a 3' block, so that when you flow in one cycle, only one nucleotide is being added. So even if you have two of the same bases next to each other, only one of them will be incorporated. And that makes it easier for the readout for the camera to take picture. So when you flow in the T with the blue dye, so the blocker is cleaved up, the fluorescent is given off. If you look at the camera at that spot, you will capture the blue spot, and the computer will translate it into a T. The next base is a G, it's green, right? So you will look at the same spot, you get the green color, and that's how you know it's a G. And the same process keeps going, and the camera just keeps taking picture. And there are millions of these dots on the flow cell, and that's how you sequence millions template at the same time. So let me show you the videos of how the bridge amplification worked.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with elan, composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub.
So Jeremy will go over the pair end sequencing assembly stuff. So basically with the Illumina, you could do single end, you could do pair end, but most people would do pair end, which as the video shows, you sequence from one end and then you sequence from the other end. The two reads will overlap. You get a little bit more information from merging the two reads. And now I think that they have 150, 250 pair end. Do you know if they have longer than that? Probably 250 pair end is probably the longest for Illumina. Now, and we are moving on to the third generation sequencing, the latest one. Well, I mean, it's come out for a while now, but it's the difference is that you could do you get a much longer read length with these. And the other thing is the third generation sequencing is a single molecule sequencing. which means you don't have to amplify the original template. The original DNA could be sequenced on the platform. And there are some benefits to that, which I will cover in a little bit. So the cool thing about PacBio technology is that they figure out how to do the sequencing in these chip that has micro wells, or these teeny tiny wells called the zero mode waveguides. And these tiny chambers are so small that they can capture the fluorescent light emitted from just one nucleotide being incorporated. And that's why you don't need to amplify the template. And by not having to amplify the template, you reduce the chances of introducing errors from amplification. So the way this works, PacBio also use fluorescent dye, similar to Illumina and the Sanger sequencing. This is the 4 different color nucleotides. This is the zero mode waveguide, the tiny chamber on the chip. The polymerase attaches, well, so when you're ready to start the sequencing, you attach the polymerase to the library. And it sits at the very bottom of the well. And every time there's an incorporation, you get a splash of color within the well, which could be captured by the camera. And this is really cool. And so when you do this PacBio sequencing, you can tell the machine how long you want to capture the movie. So it takes a movie of this splash of light, and then it interprets the sequence data from this color. And how you generate a PacBio sequencing library is slightly different from the 2nd generation sequencing library. And so one is that you need a really, really high quality DNA to start with, because now you don't want a fragment of a couple of hundred base pair anymore. You want a 20 kb, 30 kb fragment. And that means you have to start with something that has 100,000 kb when you run it on a gel. So when you shear the fragment to 10, 15, or 30 kb size, you fix the end and what you ligate it onto. So Illumina adapters are double-stranded adapter, linear. PacBio adapter, it's a single-strand hairpin adapters. It's called Smart Bell adapters. You put both adapters on the end of double-stranded DNA that it's linear. And essentially what you get after the ligation is complete is that you get a circle that it's single-stranded DNA now. And so the template is actually circular for PacBio. And then you load the polymerase onto, there's a polymerase binding site on the adapter. And what you do when you sequence with PacBio is that the polymerase will keep going rounds and rounds and rounds. The processivity of the polymerase that PacBio has engineered, it's quite amazing. You could do sequencing for 10 hours and it will keep going. And the raw read length that you get, it's quite long. It's like 50 kb or even longer. What you get is the raw read would look something like this. So you have well, actually, it should start with somewhere here. Okay, so you get a little bit of the template. You get the adapter, the other strand of template, adapter, template, adapter, template, back and forth. And the computer will remove the adapter sequence and align all the raw reads, which we call subread. So each of these is subread. They align it and then they get a consensus, which comes out to be very, very accurate. And so if you look at the accuracy of the raw read, it's quite poor. It's like 88%, which is really bad compared to Illumina, which is 99.9%. But once you turn these into a consensus sequence, the fidelity is super high. And now it's like Q50, 99.99%. Better than the short read sequencer, or at least equally good. And so this is called the high fidelity read. So you can get a high fidelity read that it's the size about 20, 30 kb. And so if you're trying to do genome assembly, imagine how much better this is compared to the 500 base pair you get from the short read sequencing platform. So just to look at pros and cons of each The short read represented by Illumina, and the long read would be represented by PacBio, but they're pretty similar. The other technologies that are short read also have similar pros and cons to Illumina as well. So the best thing about short read sequencing is that it's very cheap these days. You can sequence pretty much anything. Human genome is probably a couple $100 now. The accuracy, raw read accuracy, it's very high. Although the accuracy with PacBio, once you get the consensus sequence, it's equally good as well. So I think the accuracy could be debatable between Illumina and PacBio. The cons for Illumina is that it's GC bias. So when you get into repeat or GC rich region, it has trouble sequencing in that area. There's also some errors from the amplification step because you need to amplify the template. And the read length is very short. For PacBio, the major disadvantage is the cost. It's quite expensive. It's much more expensive than the short read. Raw read accuracy is terrible. If you don't do hi-fi sequencing, then you need a lot of sequencing depth to do the correction. Or sometimes you might need the short read technology to complement the PacBio to do the sequence correction. But there's no amplification, and so you sequence the native DNA. There's no error introduced during the amplification step. And the other good thing about the sequencing the native DNA is that you can get methylation information as well. So you can look at epigenetics if you want. There is no GC bias, and so if you need to go across repetitive regions in the genome, PacBio, it's your guy. And so hopefully you're still awake and not need the coffee yet. Jeremy is going to pick up from here and tell you what to do with these once you get the data. So after you do the sequencing, this is what you get, a bunch of ATGCs that probably doesn't make sense to you. And you have to put it through some software to put it together and figure out why you did the sequencing in the first place. And so if you have questions on sequencing, you can go ahead and ask now, because I have to go back and do another webinar. I will not be hanging around this morning. Or you can catch me later this afternoon. I'll be back for the afternoon session, but I guess only some of you will stay in the genomic group. So anybody has any questions on sequencing technologies? Otherwise, I will give this to Jeremy, and he will go over the assembly and what to do with these data.
Okay, so I've already been introduced twice, but he goes for a third time. So my name is Jeremy Shearman. I'm a senior researcher at the National Omics Center just here and an executive editor for Genomics. Okay, so I'll be talking about de novo genome assembly, the initial assembly from that, scaffolding technologies that we then use to polish the assembly. We'll talk briefly about reference-guided assembly, and then gene annotation. Okay, so I should give a few disclaimers here. So this lecture that I give, I usually give to master's and PhD students here in Thailand who have come up through the bioinformatics line. So their biology is very limited, and their computer science is their strong point. So I give a very basic background, which you are probably all familiar with. So A pairs with T using 2 hydrogen bonds. G pairs with C using 3 hydrogen bonds. Hydrogen bonds are the weakest type of bond. So G and C pair more strongly than A and T. So when you heard about the GC bias, that's because G and C pair a little bit stronger. So you all basically, the other disclaimer is that the students that I lecture to have English as a second language. So for my slides, you'll notice that they have a lot of words on them. And this is so that the students can gain all of the information. So don't learn this slide technique from me, it's bad form. Okay, so in this image here, we have each row represents one individual. And if you line up all of the sequence, you can see that it's basically the same with a few cases of variation. So you all basically have the same genome structure, and on top of that, there are some structural variations and single nucleotide polymorphisms, which gets covered in, I think, tomorrow. Okay, so the human genome is 3.2 gig, which is 3.2 billion base pairs. We have 22 autosome pairs and 1 sex pair. And you can see here's the, all of the chromosomes stained and viewed under a microscope. But each species has a different genome organization than this. So if we zoom in to one of the chromosomes, and this is actually 2 chromosomes paired up, then as you zoom in further and further, this is the DNA wound into chromatosomes. So each of these is a chromatosome, which is DNA wound around a histone. And as that unwinds, then we get to the actual ATGC structure of the DNA double helix. Okay, so when thinking about genomics and really anything to do with biology, it's best understood through the lens of evolution. So about 4 billion years ago, life began as a single cell. Roughly each generation, there's a mutation per 10 to 100 kb. And with enough generations and spatial separation, one species can mutate into 2. So all of life today can be traced back to common ancestors. And this means that the core developmental genes are shared across incredibly diverse species. And the more similar the species is, the more similar a genome it has. And this is important when we come back to reference-based assembly. Okay, so de novo assembly is the process of obtaining the genetic sequence of an organism in a way that reflects the physical structure of the DNA in that organism. And so here we have the arrangement of a fish. It has 25 chromosomes. Here's a cat. Here's a dog with 38. And a bacteria is much more simple. It has a single cyclic chromosome plus a few plasmids. So there's a lot of diversity in the size and the structure of each species. This is the current maize genome. That's corn that we grow commercially. But if you look back to their ancestor, it's very different. And here we have sorghum, the corn progenitor species, and then the completely rearranged genome of modern day commercial corn. Okay, so de novo genome assembly is actually quite difficult. And as we just saw, current sequencing methods all involve randomly fragmenting the genome. So you can imagine putting 100 copies of a book through a shredder and then trying to reassemble a single copy of that book without even knowing which page each of the pieces come from. And so the biggest problem with de novo genome assembly is repeat sequence. So to continue with the book example, imagine how many times phrases like "and then" or "so that" happen. If that happens at the start or the end, then that fragment could lead to hundreds of other fragments. Okay, so we start with a large number of copies of genomic DNA. So in this case, we're looking at a single chromosome. We start with a large number of copies. Usually we do about 50 nanograms, so that's maybe several hundred to several thousand copies of each chromosome. And then this is randomly fragmented. The most common method to fragment a genome is by sonication. So that's very high frequency sound waves that cause the water to vibrate, and the vibration then whips the fragment apart. And the average size of each fragment depends on the sequencing technology that we intend to use. So the next step is to then sequence all of these individual fragments and then put them through an assembly program which finds all of the overlaps in the sequence to generate a consensus output. So you can see here that each of the fragments has fragmented in a different location, allowing for overlaps between the fragments to be found. Okay, so the repeat sequences make it look like one read can lead to multiple other reads. And repetitive DNA can be broken down into these classes. So we have tandem repeats, which are repeats that are repeated one after the other in sequence. They consist of satellites, which are 150 to 400 base pairs, occurring in runs up to about 400 megabases. We have mini satellites, which are 10 to 100 base pairs per unit, and they repeat up to about 20 kilobases in runs. And then microsatellites, which are 1 to 10 base pairs, and you get runs of those up to about 500 bases. The other type of repeat is dispersed repeats. So these are repeats that are not repeated, but they occur often throughout the genome. So the larger of these are retrotransposons, which can be up to a few kilobases in size, although generally smaller than that, and occur up to about 800 copies in a genome. And then we have DNA transposons, which are smaller but occur far more often, up to about 1 million copies in the genome. Okay, so we saw at the start that different sequencing technologies give different read lengths. So the longer the sequencing reads are, the less of a problem these repeats are. However, the longest repeat sequences are still much longer than the longest sequencing reads. So because of this, it's important to remember that every reference genome in the database is just a model of the actual genome and may and likely does contain errors. Okay, so to do the actual assembly, there are two main algorithms that are used. There's overlap layout consensus and something called a De Bruin graph. So overlap layout consensus, which we call OLC, the idea is to construct overlaps directly from the read using the entire read. And this algorithm was designed for long read lengths. The first of these was the Celera assembler, which was designed and used to assemble the human genome back in 2001. The other is a De Bruin graph. So the idea here is to break each read down into smaller fragments, which we call a kema, and the original read is then discarded, and then the kemas are aligned and assembled. So this method was designed for short read technologies, which means that repeat sequences are highly problematic. Okay, so assembly programs get updated very frequently as sequencing methods and computing power increase. So the programs that I've listed here may or may not be up to date by the time you actually come to use them. I think in my 12 years of assembling genomes, I've never used the same assembly program twice. So OLC, we have Celera, which was used for the human genome, and then Canu, HGAP, and Falcon, which are actually derivatives of Celera. De Bruin graph programs, which are fast and loose. We have SOAP de Novo, Abyss, Paths, and Velvet. These are just a few of the more popular examples. There are tens or hundreds of programs out there. There's also some assembly which combines the two, so, such as Ray or Spades. So the first start for those is to use a De Bruin graph, but instead of discarding the reads are kept and then mapped back to the resulting contigs. Okay, so this is what a kema is. So if this was the initial segment of DNA, and we do short read sequencing of that, we get each of these shorter overlapping reads. Just have a drink of water, 1 second. Okay, so we have these shorter overlapping reads, and you can see how they overlap here. The idea then is to break these down further into much shorter fragments, which sounds counterintuitive considering how problematic repeat sequences are, but it seems to work pretty well. It's a trade-off between computing time and accuracy. And so then each of these kemas are aligned, which produces this De Bruin graph here. And so you can see kemas that have one in and one out in the graph are used to essentially scaffold the rest of the kemas. The program then chooses the path that uses the most number of unique kemas to link each of the solid kemas, and this is then collapsed down into a single sequence. So selecting the right kema size is important. Larger kemas compute more slowly, while smaller kemas have a higher chance for error. So it's a trade-off between accuracy and time. So when De Bruin graph first came out, was about 2005, 2006. High performance computing was not as common then as it is now. So I think a lot of universities didn't even have a high performance computing cluster. So the ideal kema size depends on the complexity of the genome that you're looking at, and there are optimizer programs that can choose the size that gives the highest number of unique kemas. But in general, larger kemas generate a better genome assembly. Okay, so the other algorithm is OLC. So this consists of 3 steps. So here in this example, we have 2 input sequences, and you can see in red, this represents a repeat sequence. So this sequence is identical to this, and then the green, blue, yellow, and orange are unique sequences. So the first step here is to line up all of the reads to find the overlaps. This is then laid out into a graph showing the overlap of each of the reads. And this is then collapsed into single sequences. So you'll notice the single sequences are called contigs. So you'll notice that even though we essentially had 6 input sections, the output is 5 contigs. OLC is very resource intensive. So because each read is aligned to each other read, any additional read that you add exponentially increases the computing cost. Okay, so this is just a summary of the kind of output that the assembly algorithms give. So we have OLC, which actually can output overlaps in the repeat sequences. We have the De Bruijn graph, which tends to give a clean cut in the contigs where there are branches in the graph. And we have string graph, which is essentially a version of OLC that gives the same clean cut at repeat sequences that the De Bruijn graph gives. Okay, so all of these algorithms return back a set of contigs. Repeats that are too large result in multiple contigs. Longer reads result in fewer longer contigs, and shorter reads tend to give you a much larger number of short contigs. So, for example, we've sequenced the rubber tree genome. It has a haploid size, so that's 1 copy of each chromosome, of 2.15 gig. And we sequenced this using only Illumina. And the result from that was 1.2 million contigs. And if you sum up the size of those contigs, it was 1.1 gig, which means that approximately 1 gig of sequence represents repeats that have been collapsed into a single contig. And we've also sequenced the sugarcane genome. The entire genome is about 10 gig, and it's an 8 to 12 ploid, which means that instead of having 2 copies of each chromosome, it has 8 to 12 copies. So we sequenced this using PacBio only, and the resulting assembly produced 104,000 contigs, totaling 7 gig. So the reason that such a larger amount of the genome was contained within the assembly is because of the long PacBio reads able to span across the repeat sequence and link repeat sequence with unique sequence. So the repeat sequences that can't be spanned get collapsed into single contigs. Okay, so once we have a set of contigs, the next step is to try and figure out how they link up. And this stage is called scaffolding. So one of the initial methods was to use long-range paired end reads. So if you remember back from Illumina, the sequence is attached to the slide, and then with bridge amplification, you get the other side of the sequence. So with long-range paired end reads, you fractionate the DNA so that you get an average of 4, 8, or 20 kilobases. Usually you do 1 library of each. And then you get a read from the start of each fragment and a read from the end of each fragment. And because you know roughly the average size between those, you can say that those 2 reads are 20 kilobases apart. So if you map those reads then to the resulting contigs, and 1 read maps to this contig, while the other read maps to this one, then you know that there's about 20 kilobases of sequence in the middle, which is likely to be a repeat. And so by doing that for the whole genome, you can essentially scaffold each of the contigs into a larger sequence that's separated by ends to represent the repeat sequences. One of the latest and so far best approaches is called High-C, which stands for high throughput, chromosome conformation capture. So although pretty much every time you see a chromosome, it looks like this, this is actually almost never what the chromosomes look like inside the cell. So this is the chromosomes that are tightly packed for mitosis, very tightly coiled around the centromeres. And in that state, transcription is not possible because none of the proteins in the cellular machinery can access any of the genes. So what the structure of DNA inside the nucleus looks like most of the time is this, where each of the chromosomes are unwound and they're largely localized. So in this image, each color represents one chromosome. And this is to allow for all of the transcription and translation machinery, all of the promoters and repressors that have access to the DNA. So Hi-C was originally developed to identify distal parts of DNA that are brought into close proximity to bring related genes together or to allow distal enhancers or insulators to function. So the way that this process works is chromatin is cross-linked using formaldehyde, and then the DNA is digested using a restriction enzyme. The DNA around the coiled chromatin is protected, and DNA fragments that are closer together will ligate together more often, which results in a statistical problem. Okay, so the cross-linking looks like this. So we have the amine group on a protein. The formaldehyde then attacks this to produce a methylol known as a shift base, and then the shift base can interact with a nucleophile on a DNA or a protein, resulting in a chemical link between either two proteins or a DNA and a protein. And so this process gets applied to whole nucleosomes, to an entire nucleus of a cell. And so it can produce quite a mess, with each piece of DNA being able to link to another piece of DNA or to a histone or to any other kind of protein that's in there. So the way to get reliable results is to use a concentration of formaldehyde that's high enough to produce a large number of links, but not so high that everything just gets linked with everything else. So the idea is that two histones come into contact. The formaldehyde then links them together. It links the DNA that's around the histones to the histone. And then when we use a restriction enzyme to cut all of the DNA, we have left some DNA attached to a histone that's attached to hopefully a nearby histone that's attached to some DNA. The ends of this then get marked with biotin. They get ligated so that the DNA actually joins together. And then this DNA gets sheared, allowing us to isolate the biotin-labeled DNA, which then gets sequenced using paired-end sequencing. And so here we can see just a heat map of chromosome 14. And so this says that the DNA at this part binds often to DNA here, and sometimes the DNA that's very, very far removed from its actual physical location inside the nucleus. And you can see that here we have the Hindi3 restriction enzyme, and this is NCOL restriction enzyme. So different restriction enzymes give a largely similar but somewhat different output. So the statistical problem that I mentioned, about 10% of the time, DNA that's found linked together by this method will be less than 10 kilobases apart. About 15% of the time, it will be within 10 to 100 kilobases. About 18% within 1 meg, 13% within 10 meg, and 21% of the time, the DNA will actually come from completely different chromosomes. So it looks like a mess, but combined with PacBio length assemblies, research groups are reporting around 1 to 3 contigs per chromosome for de novo assembly of large complex genomes, such as humans, goat, and rice. Okay, so the next part is reference-guided assembly. So the assembly part of this is the same as de novo assembly, but instead of scaffolding with high C, we scaffold with a reference genome. So this was primarily designed for what we call resequencing. So it's when there's a reference genome that exists, but you want to sequence a different individual than what that reference was. So the idea, we have contigs, we have the published reference genome, we can then align the contigs against the reference genome and produce a scaffold of these contigs. So this is done by looking for a seed hit within the contig against the reference. And once a seed hit is found, the alignment program then extends out from that. And the result is, so here we have one single contig aligned against the reference sequence. And you can see that there are some differences in length. So that would represent maybe insertions or deletions between the reference and the individual that we've newly sequenced. So this method can also be used for very closely related species if you have large enough contigs and you can properly validate the output. So validating the output is done by mapping the scaffolds that you produced back against the reference and plotting it. So in this case, we have the newly scaffolded genome against the reference, and you can see that there's a large inversion here. So if the two species are similar enough, then you'll get a nice plot similar to this. If the two species are too separate, if they're too different, then the result will be a mess, and you'll see sequence from your scaffold split up and coming from a large number of different chromosomes. So if you try and publish this kind of thing, you need to start with very large contigs, and you need to properly validate the output. Otherwise, it'll just get rejected. Okay, so the next step is genome annotation. So this is following on from, so here we've sequenced the genome, we've scaffolded it hopefully into complete chromosomes. The next step is to identify and list genes. So for here, we identify genes, repeats, ribosomal RNA, transfer RNA, and messenger RNA, and output the location within the genome. So there are various ways of doing this. including gene prediction based on sequence and knowledge of start-stop codons and splice sites, RNA sequence data, including de novo transcriptome assembly, and protein information of highly related species mapped to the translated genome that we just assembled. And the best approach is to combine all of these methods into a single output. And there are two currently popular pipelines to do this called Maker and Evidence Modeler. And my lab uses Evidence Modeler. Okay, so firstly, what is a gene? So a gene is a basic physical and functional unit of heredity. For a long time, it was considered that one gene produced one protein, which gave one function. But later we identified alternate splicing and protein moonlighting. So this became one gene can give 1 to 5 proteins and equal 2 to 10 functions. And then later we found functional RNA. So now we have one gene can give 0 proteins and have one function. But we still generally focus on the one gene it gives 1 or more proteins definition. And the main reason for this is because proving that RNA is functional is very difficult. It's something that has to be done with a lot of lab work. And so when we're assembling an entire genome, nobody has the time to go and identify all of the functional RNAs within it. Okay, so this is the pipeline. So we've sequenced our genomic DNA. We've assembled all of those reads. And in this example, we're using Kenu because we used PacBio. So we have long reads. The next step is to mask all of the repetitive sequence. Okay, so we do this using a program called Repeat Masker, which basically just scans through the entire genome to identify all of the repeat sequences that I talked about earlier. So it identifies transcription elements and compares against a repeat database. Okay, so the main reason for doing this is to mask out the transposable elements. So these are the direct repeats. So these originate from viruses and bacteria, and they're often referred to as jumping genes because they contain genes that transcribe them, and then they can move to a different part of the genome, get reinserted, and then you get an extra copy. So here we can see a transposon, a long terminal repeat transposon, a non-LTR retrotransposon, and this is another kind of transposon. There are even some viruses that are still currently doing this, such as the herpes simplex virus, which inserts itself into the host genome. So transposable elements is a process that's ongoing still. So the reason that we have to do this is because this can exist hundreds of thousands to millions of times in the genome, and each copy here has 3 to 4 genes. The resulting gene annotation file would have several million genes, most of which would just be virus. So the output of repeat masking is to just replace all of the repeat sequence with ends. And that means that when we put it through the programs later, no genes can be identified in these regions. Okay, so next is RNA-seq. So RNA-seq is basically the same as DNA sequencing, but it's performed on cDNA. So cDNA stands for coding DNA. So if we start at DNA, we have exons and introns. This gets transcribed to RNA, and the introns are excised out to produce the messenger RNA. So to make cDNA, we then use the messenger RNA as a template and transcribe it into DNA, giving us double-stranded DNA that represents this messenger RNA. And this is then sequenced on usually a short-read sequencer like Illumina, and then we can map the resulting sequence back to the genes that they were transcribed from. So we use RNA-seq to generate a transcriptome assembly, and the best way to do this is to align the sequence to the genome using a splice-aware alignment and assembly program. And in my lab, we use parser for this. So this type of assembly is better than a no-reference de novo assembly, because that type of assembly can mix genes that are similar in sequence, but separated in the genome. So proteins tend to have a lot of similarity between their physical structures, and this can cause a lot of problems in a no-reference de novo transcriptome assembly. A benefit of parser is that it can also identify alternate splicing, where some de novo assemblers call them as separate genes. So the result from this is an entire set of genes, including exon locations based on the RNA-seq data. So the organ of choice for RNA extraction for this is important here, because different cell types each generate a different subset of the genes. So it's best to mix multiple organ types when you're generating the RNA-seq library. So for humans or for animals, you'd mix muscle, heart, brain, blood, and skin. And for plants, you'd want to mix flower, leaf, root, and meristem. We also use the RNA mapping result as an input for evidence modeler later, and we use the program GMAP for this, but there are many others. Okay, so the result of this output is in a data structure called a general feature format, or a GFF. So all of the data that comes out of the field of genomics is so large that the only way to actually look at it is through some kind of program. So because of this, we need the output data to be machine readable. And the best way to do this is to have consistent data structures across different species. So every genome annotation file that you download will be in this GFF format. So the GFF format is essentially a table of 9 columns. The first column is the name of the sequence. The second column is the source, which can have information about how this sequence was generated, such as Augustus or repeat masker, for example. The third column is the feature. So this says what the sequence is. So it could be a gene or an exon. Column 4 and 5 is the start and end. 6 is the confidence score of the sequence. 7 is the strand, so whether it's on the plus or the minus strand of the genome. And then we have phase, which is usually not used for very much, and attributes, which is usually used for the name of the gene. So a GFF looks like this. So here we have the name, CTG123. This particular sequence is a gene. It starts at base pair 1000, goes to base pair 9000. It's on the plus strand of the sequence, and the gene is named gene 00001. So most programs just assign a gene number. Okay, another method is to align proteins from highly related species. So here you usually use a handful of highly related species, download all of the protein data that exists for that, and then map it against your assembled genome. So here we have our assembled genome sequence, and each of the possible reading frames for the plus strand, each of the possible readings frames for the negative strand, and then the protein sequence of related species is then just compared to each of these to look for overlaps. So our lab uses a program called AAT for this, and it can compare a genome to a protein database or a cDNA database. And similar to parser, the output of this is a set of genes with exon locations in the genome. So a consensus is taken when multiple proteins and cDNAs are used. So here we can see we have the query, which is our genome, and cDNA and protein sequences from a bunch of related species. Okay, so the third method for gene identification that I mentioned was ab initio gene prediction. So ab initio gene prediction is the application of everything that we've learned experimentally about what makes a gene. So start and stop codons, the statistics of, or the statistical probability of an open reading frame, exon structure, splice sites, and promoter and enhancer sequences. Okay, so you probably all know this. We have 64 codons. The start codon is AUG, and there are 3 stop codons. So the probability, if DNA was random, that you would get an amino acid sequence larger than 20 starts to decrease the longer you get. So we can generally predict that any amino acid sequence larger than 20 is unlikely and therefore more likely to be a gene than random chance. This is based on the human genome. And in reality, most sequence is not random, but still large open reading frames are uncommon and pretty good evidence that we're dealing with a gene or a pseudogene. Okay, so exons are coding segments of a gene that are separated by non-coding segments, which are introns. Each exon has a splice site. You can see here, it's usually AG followed by a GT. Then roughly 10 to 10,000 base pairs away, we have a branch site. And at the start of the next exon, we have the splice acceptor site. So that's an AG in the intron, and then a G or an A in the exon. And so inside the cell, when a messenger RNA is being processed, the intron is cut out and the exons are stitched together to produce the final messenger RNA. We also know the sequence of the most common promoters and enhancers. So we have the GC box, which is usually about 100 base pairs upstream of the start. We have what's known as a cap box, and we have the Tata box. So the presence of these sequences are also good indicators that there's a gene present. Okay, so ab initio gene prediction just applies all of that, and the output of this is a set of genes in GFF format. Okay, so we've now done this step, and this step, but that's produced 3 separate GFF files. So the next step is to merge those 3 files into a single file to produce a weighted consensus gene annotation file. So because there might be inconsistencies between those, we apply weights based on the relevance of the input data that we used. So we give the highest weight to parser because that is the RNA-seq transcriptome assembly. So this is actual empirical evidence that we have that says that there's a gene there, followed by GMAP, which is the RNA-seq mapped against our genome, followed by AAT and Augustus equally, because these are just predictions. So we expect about 20 to 40,000 genes for your average species. Okay, so now we have the genome assembled. We've scaffolded it into chromosome-level scaffolds. We've identified where the genes are. Now we need to try and figure out what these genes do. So the GFF that we get back has only this information. It says there's a gene here. It has this completely useless gene name, and that's all we know about it. So to assign a function to each of these genes, we use a program called BLAST2Go. It started out as a free program, but it's now been packaged into a commercial program called the Omicsbox. So the idea here is to map each of the genes against a database and then identify what the gene does based on our information about the genes in the database. So the first step is to blast and map, and then we identify the gene ontology. We use Interpro to identify any known protein domains, and then use it, get the enzyme code, and the last step is to get its location in any of the PEG pathways. Although depending on the species that you sequence, that tends not to give much of an output. Okay, so Interpro provides functional analysis of proteins by classifying them into families and predicting protein domains and important sites. The Interpro motifs are directly queried at the Interpro scan website by the program. For EC numbers, these specify enzyme catalyzed reactions. So the EC number is actually for the reaction that the protein is involved with, not the actual protein itself. And so because of this, convergent evolution means that proteins that are actually quite different can end up with the same EC number. So BLAST2Go has an EC annotation file which is mapped to the proteins that you input. This is an example of PEG. So here we have all of the various genes, and this one is for the chemokine signaling pathway. So KEG is just a series of pathways like this where we've experimentally validated each of the genes that take part in it. So when you're running BLAST2Go, you start with a sequence name that's been output by the program, and the result is a description of what the gene actually does. So in this example, it was gene 4, and it's an endochitinase EP3-like gene. Okay, so the final step we've assembled the genome, we've got scaffolds, we've identified genes, we've annotated those genes with a function. Now, before we publish, we want to know how good our final assembly and annotation is. And so this is done using a program called BUSCO, which stands for Benchmarking Universal Serial Copy Orthologs. So this is a set of genes that occur in almost every species. So remembering back to my evolution slide, a lot of species have a set of genes that are present, that are almost always present. And so if we compare our annotation to these BUSCO gene set, then we can get an idea of how complete the genome is. So sometimes some of these genes are lost or gained, so we don't expect all of them to necessarily be present, but anything in the 95% range is considered a quite complete genome assembly. And there are ortholog databases that exist for bacteria, archaea, eukaryotes, fungi, and plants. Okay, so that's the final summary of genome annotation. Any questions? I think I finished much earlier than I anticipated.